Hey y'all, welcome back to Dry
Infinity.
Infinity. Today I'm here with
Anniversary.
and Viva. Today we're going to be learning about epigenetics. Let's get started.
So, let's start from the very beginning. So we're going to talk
Okay.
about transcriptional factors, right?
Yeah.
So let's start that. So, hang on a second, bear with me. Okay.
Zone in.
So, transcriptional factors. In eukaryotes, transcription of target genes can be stimulated or inhibited when specific transcriptional factors move from the cytoplasm or the nucleus into the nucleus, right? So this can turn on or off genes. So only certain proteins are produced in a particular cell. So turning off particular genes in the cell is what enables them to become specialized. So we're talking about in eukaryotes. So this is to do with animal cells or in our cells, because we have eukaryotic cells as we are animals. The transcription of these target genes can be stimulated, i.e. progressed, or stopped, okay? When specific, so only certain transcriptional factors will be able to move from the cytoplasm into the nucleus. So we're going to move on to transcriptional factors in a minute. We all know exactly what they are. So just think of them as a molecule at the moment. So they basically stimulate or inhibit target genes. And target genes are basically to do with the chromosome. So we have our chromosome over here, right? And then we've got certain genes on our chromosome that we want to target for transcription. So this can turn on or off genes. So it can basically i.e. stimulate or inhibit the genes. So only certain proteins are produced in a particular cell. So we're turning on or off genes because we want to create certain proteins. So we only need one gene for each protein, I suppose. So we can put a gene equals one protein. So turning on or off particular genes can become specialized, allows the genes to become specialized, so particular to one function. Right. Okay, so we're going to move on to transcriptional factors now. The transcription of a gene will only occur when a molecule from the cytoplasm enters the nucleus. So we need to remember that, and binds to the DNA in the nucleus, okay? These molecules are called proteins, and these are basically transcriptional factors, and each one can bind to different base sequences on the DNA. And this can initiate transcription of the genes. Sorry.
Beep, 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 time is up.
Of genes. So we've got our DNA binding sites. So this is on the DNA molecule, we have a binding site, and we also have some receptors there. So this is outside in the cytoplasm. And once this transcriptional factor, i.e. a protein, so a protein is going to be here, once it binds to the DNA binding site, they will enable the receptor to bind to different base sequences on the DNA. And this will allow transcription. So what I mean by base sequences, which is basically just like...
I need a flippin' wee.
On the DNA molecule, we have A, C, E, G. You can basically bind to those bases. Once bound, transcription begins, creating the mRNA molecule for that gene, which can be translated into the cytoplasm. Oh, I did too much highlighting there. With me. They can be translated into the cytoplasm to create the protein. So now we're talking about when the protein binds to the DNA binding site, and basically what we just said before, we're basically creating the mRNA molecule once it's bind, and then we can translate it into the cytoplasm to create the protein. So we're basically creating the template mRNA molecule after transcription begins, of course. So without the binding of a transcription factor, the gene is inactive. So if we don't have any binding factors, it's really good for your essays. If you do have stuff on proteins and stuff, because transcription factors are to do with proteins. So without these proteins, we won't be able to make the genes active, and the protein won't be made. So we won't be able to make proteins without no transcription factors, and that's absolutely crazy, because just having no proteins such as estrogen, which is what we're going to talk about in a minute, if we don't have estrogen in the body, which are very small chemicals, we can't even see them, we won't basically make any proteins, and we won't have any basic building blocks in our body like enzymes. We won't be able to create those. We won't be able to create anything else, like we've got protein channels, so etc. We can move that onto anything. So we can talk about that first in our essays. Sorry guys, I didn't mean to say ass. So estrogen is estrogen is a steroid hormone that can initiate transcription. So assistant, do you know what steroid means?
Yes, people take it to gain muscles.
Right. Yes, this is actually correctish in a way.
Thank you.
It basically, steroids are just a thing, so they can initiate transcription. Okay. So what I'm basically saying is that estrogen can initiate transcription because it's a transcription factor, and it does this by binding to a receptor site on the transcriptional factor. So we're talking about the receptor site here, like we mentioned earlier. So this is the binding site. Oops, sorry, wrong part. And this is the receptor site here. So once it enters the cell surface membrane, so you can see estrogen is basically entering the cell surface membrane. Sorry guys, I'm really ill. And then so that means that estrogen must be polar. Must be non-polar, sorry, because it can enter through the cell surface membrane without protein. So that's one thing you can link it to. So we can talk about it binding to a receptor, and then we've got the transcriptional factor over here. It does this by binding to a receptor site, which is here, on the transcriptional factor. The transcriptional factor is here, yes, all this is the transcriptional factor. So make sure you know that. All of that is the transcriptional factor. When it binds to the transcriptional factor, it causes it to change shape slightly. So when it basically binds, the transcriptional factor will basically change shape.
Beat Dolan.
So the transcriptional factor is what I circled here. So this can change the shape slightly, and it makes it complementary to the DNA bases, which is down here. Okay. So this will then initiate transcription. And yes, of course, it highlights everything at the end. So, yes. So what we're talking about, if this did not make sense to you, I'm going to explain it again. Only because I messed up a bit. So I'm going to erase everything.
Dolan.
So we're going to go through the whole process. My assistant is being very, very inappropriate.
Sorry about that.
So, so estrogen is a transcriptional factor. Okay?
Mm-hmm.
And it's also non-polar. So I'm just going to put non-P for now. And it basically enters through the cell surface membrane, and then it binds to the receptor site, which is here. I am so sorry. Estrogen is not the transcriptional factor. Oh my goodness me.
Excuse me?
Fucking hell.
No swearing in this classroom.
This is the shitty transcription.
Thank you.
Alright. Okay, that's the transcriptional factor. So that binds to here. And that changes shape. Because you can see there's actually two. There now. And that with the receptor is still attached to it. And then it will basically move into the DNA to initiate transcription. So this is the promoter region. They did not mention it here. So PROMO, promoter region, which is basically what promotes the transcription, basically. Okay, so to summarize what I said was that we have transcriptional factors. Okay, they can stimulate or inhibit transcriptional factors, and those can turn on or off genes. Turning off specific genes and on genes is basically making specific proteins, which means that it enables them to become specialized because it's particular to one function. Because proteins can't be non-specialized, can they? Because they are specific to different sort of regions of the body. Like you've got hemoglobin, which is for binding and loading and unloading oxygen. We've got like ribosomes, which is making more proteins. It's crazy. So yeah.
Thank you.
So now we're moving on to transcription factors. Basically, transcription of a gene will only occur when one molecule from the cytoplasm enters the nucleus. Okay.
Mm-hmm.
So one molecule of cytoplasm enters the nucleus. This will bind to the DNA. Okay. So it's basically telling you a simple, nice sentence about how a molecule from the cytoplasm, which is like estrogen, and this will enter the nucleus. Remember, we said it will bind to the DNA. So these molecules are protein transcription factors. It's going to be bind to different base sequence of DNA, and this will initiate transcription. It's basically giving you an overview. Once bound transcription begins, creating the mRNA molecule for that gene. So when it's binded, the transcriptional factor, it will basically create the mRNA molecule, and that can be translated in the cytoplasm to create the protein. So without transcriptional factors, it's basically saying the gene is inactive and the protein won't be made. How sad. So estrogen. So this is the example that we have to use. Estrogen is a non-polar molecule. It could diffuse through the cell surface membrane. And what it will do, it will basically bind to the receptor. The receptor will then binding to a receptor site on the transcription factor. So the receptors are obviously, well, in this visual image, it's not really great, is it? It just looks like a line, but all that is one molecule, okay? So that binds to the receptors, one molecule, and then that will change shape of the transcriptional factor. When it binds to the transcriptional factor, it causes it to change shape slightly change the shape. So that will then enable bases to of the DNA to bind to the transcriptional factors. Okay. Let's say making it complementary. Remember using the word complementary. Make sure you use the word complementary. And that enables it to bind to the DNA to initiate transcription. Okay, and we're done here.
Yay. Well done, everybody.
That was our next subsection that we've done. Perfect.
Okay.
I'll see you guys later. Bye.
Bye, y'all.